Hello and welcome to the Snaps
Okay.
with Dr. Otito, where we spotlight diverse voices and actively foster collaboration. I'm your host, Dr. Kimbo Otitoji, and this is our very first episode. I'm so excited. Here on the Synapse, we bring together brilliant minds across the world to share insights that shape research, technology, and innovation, especially from African perspective. And today we are beginning with one of the most transformative fields in modern science, which is bioinformatics. My guest today embodies the new generation of scientists driving change. She is a research scientist at the National Biotechnology Development Agency. I'm a postgraduate researcher whose work focuses on bioinformatics and computational biology. She is also an accomplished scholar with multiple publications in reputable journals, contributing to the growing body of African-led bioinformatics research. Please join me in welcoming Dofan, who you're welcome. Hello, Dofan. How are you doing this morning?
Thank you, Dr. Akin. I'm fine. Dr. Akin, you're way too kind. Thank you very much.
All right. Thank you for accepting to be my very first guest. And so let's begin. Can you tell us
I'm
briefly?
honored to be here.
Thanks. So can you tell us briefly about your journey? What inspired your interest in science generally and now bioinformatics?
Okay. I've always loved science as a child. I love the science has always been what I've loved right from when I was growing. My favorite subjects were science. Science has always been interesting to me. And then how I ventured into bioinformatics, it was not by design. Somehow I found myself into it because of the boss I had at the time. And luckily, I've always also had interest in computers. So it was like a double treat for me, merging science and my love for computers. So we picked from there.
Okay, that's good. And now that
Yeah.
you are pursuing your postgraduate studies, can you tell me about your
Yeah.
work and its relationship with what you do at the National Biotechnology Development Agency?
Okay, so my work currently, we are looking at AMR, that is antimicrobial resistance in highly infectious diseases, tuberculosis to be exact. AMR is a scourge. So we are looking at bringing up new drugs, like my postgraduate work. I'm working on a hybrid compound, and all this we are doing it computationally. We are trying to merge peptides with actual small compounds. We want to create a new hybrid compound that uses the penetrative power of peptides and the therapeutic power of biochemicals or small bioactive compounds in plants, to target AMR in tuberculosis, because the frontliners in tuberculosis are no longer working,
Hmm.
and that's an issue. The second liners are too powerful, so there's an issue with treatment in TB. We are having extra drug resistance. We are having multi-drug resistance. So that is what we are trying to address.
Okay, thank you very much. So, in line with our team, which is building Pan-African bioinformatics capacity to bridge the biomedical divide, and I'm glad to see that you are
Yeah.
working in the field of bioinformatics, where you are using computational tools to solve medical challenges. However, for the purpose of students who may come across this program, maybe we should uh, take a few steps back and ask ourselves, what exactly is bioinformatics? And then quickly, maybe a two-in-one question, use your area of your current research, all right, to probably walk us through a typical workflow, okay, from data collection to discovery.
Okay. So bioinformatics, in very layman's terms, is the use of informatics, that's computer knowledge, to be able to analyze, store data, biological data to be precise. So, and bioinformatics is very wide. You know, we have so many branches of bioinformatics. We have the genomic side where you are dealing with DNA, you are dealing with RNA and all that. And you still have proteomics where you are dealing with proteins and all that. But for me, I am more into pharmacoinformatics, which is the branch that deals with the computational drug discovery.
Okay.
So for my work, like you asked, because you are not going to the wet lab to get any data, you rely heavily on data that is found in databases. People who have done work over the years, we have the advantage. They've been able to store most of this data in various databases, and these databases are highly specific. You know, we have protein databases, we have for chemicals, we have for DNA, we have for sequences and all that. So it depends on what you're working on. You go for the database that you're looking for. As for me, going from my own research, I needed to the small compounds that I'm working with, all these were gotten from literature. And when you get it from literature, you rely heavily on literature because, of course, you're not going to the lab yourself. So, you know, you are able to save time and you are able to tap into the wealth of other people's knowledge that is available, readily available out there. So we create a library of compounds. Most times you can have a very specified library. That all depends on you. As for me, I just profiled most edible plants and medicinal plants found here, indigenous to Nigeria. That is why I used to build my own library of compounds. And we built that and we were able to use databases to get the information or the physicochemical properties of this, what do you call it, of these bioactive compounds. And also because I was working with peptides as well, I was working with antimicrobial peptides. I also got that online. That information was all gotten from literature, you know, because there was no wet lab. And that's the beauty of bioinformatics. So by the time you're able to get all this data online, you know, you do a lot of in-depth, you do a lot of review, you do a lot of research. Even if you are not going to the wet lab, you can't just carry any data you see and just run with it. You have to filter, you have to go through and confirm that this data is actually accurate, you know, based on all that. So when we have this data, we use softwares, you now start your analysis. This usually takes, depending on the size of your library, if you have more compounds to work with, you could spend like up to a month running, you know, the entire process of finding out the drug-like properties of compounds, especially if if the target is to combat a certain therapeutic challenge. Okay, like for me, like I said, we're trying to look at AMR to tackle AMR in what they call it, tuberculosis. So that took me about 2 months, yes, from data collection, from the point of data collection to analysis and verification. All your information is based on other people's work. We don't call it, you don't report your results in absolute. Your results come in predictions. You know, you say you predict, you predict, you predict. So where you get your absolute is this just shortens your lab time. So instead of the time I would have gone to go and be picking plants in the bush or wherever to come and analyze and screen. I've crossed all that. So now I'm going to the lab with certain compounds to work with. So that just reduces your lab time to
Oh,
more
okay.
than half the time.
Okay. So I think that takes me to my next question. So that means there is a way by which you can make your data generated to be biologically meaningful, such that
Yes.
they are not data noises.
Yes.
Okay. By actually working with wet labs.
Yes.
Okay. I know you are well published in peer-reviewed journals. So can you share with us a study that you are most proud of?
Yes. The study I'm most proud of was when the COVID-19 pandemic hit us in 2020, and there was a lockdown. There was so much fear, and everyone was wondering, what do we do? There's no cure and all that. And so, under the leadership of our boss, of our, how I put it, our divisional head at the time, Dr. Ruwai, we got different targets on the coronavirus itself, and we did a computational study, to find out compounds that could target the COVID-19. I was very proud of that work because we came out with results. I think we even submitted our results to the Ministry of Health at the time, but we never heard back. So, but that was one of my proudest work. We wrote about 4 papers
Wow.
on that, and I'm really proud of that work.
Okay. I know that some students are scared of coding. Some are scared of statistics. Now, but they are interested in bioinformatics. What would you say to them?
Um, for myself, I- I've I've been doing bioinformatics now for the past 8 years. I am not proficient in coding. I am not proficient in statistics.
Okay.
All you need to do is to know how to learn. When coding, when you're interested in coding is when you want to do back end. And what do I mean by back end? If you are interested in actually creating new pipelines, there are so many what do you call them? So many tools for you to run your analysis. You don't need to be a coder, you don't need to be a statistician, and thank God with new AI tools that are coming in now, which could even do that. If you even need code to code, you can always ask many AI tools to help you code, but you don't really need coding. I will say programming language like Python is important. I'm on it myself, but you don't. It's not a prerequisite. You can always go into that without having coding knowledge or
Oh, thank you.
deep statistical knowledge. Yeah.
All right. You touched on AI. So considering the advent of AI and big data integration, where do you see bioinformatics evolving over the next 10 years from now?
Oh, it's in fact AI is so powerful, and AI and the integration of machine learning, they are beginning to integrate these things into most of most processes now are being automated. Like something like building a library, how you had to go manually to do searches through search engines and all that. People, through the help of AI tools, are using SQL to build libraries of thousands and thousands of compounds in a very short period of time. So AI, AI on itself cannot it's not powerful. It is you, the person using the AI. It's what you can do with the AI that matters. AI is a tool. It's what you make with it that matters. So I will encourage everyone. There's a saying that they say AI will replace people. That's not true. AI is not AI that will replace people. It's people who can use AI that will replace the people who can't use AI. AI is powerful if you know how to use it. I will encourage everyone, irrespective of what you are doing, irrespective of your discipline, learn AI and integrate it into whatever you are doing. You'll be unstoppable.
All right. Thank you very much. Now, what is the role of collaboration in the advancement of bioinformatics?
Yeah, collaborations are important because not only are we, you know, while knowledge could be universal, especially now with the advent of the internet, sometimes we still have a little cultural context. So I would say like how I may learn something might be different from the way somebody is learning it in the US and all that. So by the time you are rubbing minds with people or in industries, across industries. And what I mean across industries, I'm not talking of disciplines per se. I'm talking of like I'm in a research institute and I'm collaborating with somebody who is in the academic space, how to approach their work. So by the time you collaborate with these people, you see you're able to share ideas and research. It makes research more robust, and I think that's the beauty of research, especially by the time you start reaching out, learning from other people, collaborating and bringing out very meaningful research.
All right. Thank you. So on a lighter note, if you weren't a scientist,
Okay.
what other career might you have pursued?
I don't know, probably an investigative journalist.
So,
But
this has been an inspiring conversation. We've explored how bioinformatics is helping to decode the language of life and how African researchers are contributing to global discovery through data-driven science. But before we wrap up, in one sentence, what is your message to young scientists like myself watching the house today?
Dr. Akin may be young, but you are not a juvenile in the science world, but all the same, and we still answer to the young scientists. Um, don't wait, don't wait. You know, science is growing, science is evolving, science is so dynamic. It's like when you just blink, something new comes in. So don't wait. If you have an interest, pursue it. And for myself, I took a very long break. I took a very long break from science because of the Nigerian factor, where I found myself, where I got a job after my youth service. And so that set me back a while. But I
Okay.
always knew science was what I wanted. So if I will urge you, most times after your first degree, if you can just continue, you know, have a solid interest. Don't just go into science for the sake of reading science. Science pays. People just say school is scam, but it's not. It's application. You just need to know how to apply it. And with information these days, YouTube is a very powerful resource. You can always use YouTube. You can even use AI to ask, what can I do with this? How can I make meaning of what I have? You know, nothing is useless. No knowledge is useless. If you have an interest in something, pursue it with all your heart and be the best at it.
Wow, beautifully said. If you have an interest in something, pursue it with all your heart. Use every means available. Our guest today, Dofan, has encouraged young scientists to even go through the school of YouTube. This is wonderful. So on behalf of the Synapse team, thank you, Dofan, for joining us today and for the important work that you are doing in advancing scientific innovation through bioinformatics. To our viewers, thank you for tuning in to our very first episode. Join us again next week for another exciting conversation that bridges science, society, and the stories behind discovery. Once again, thank you, Dofan. It was nice having you on the show today. I am your host, Dr. Akimbobola Otitoju, signing off from the Synapse. Until next time, stay curious.
Thank you very much for having me.
Thank you, thank you, thank you. Until I see you
Thank you very
again
much for having
next
me.
week,
Thank you.
same time, stay curious, stay inspired, and keep thinking beyond the box. Thank you. Bye for now.
Bye-bye.